Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this lecture, I will be going over cholesterol biosynthesis. Now let's get into the details. Cholesterol synthesized by almost all cells of our body, but most of the cholesterol biosynthesis will occur in the liver cells, and also a significant quantities of cholesterol is synthesized in the intestine, kidney, gonads, and also in the placenta. Almost all cells in our body needs cholesterol, and the cholesterol that is there in the plasma, which is transported from one organ to another organ, it is present in the form of a cholesterol ester. So about 2/3 of the cholesterol in the plasma, it will be present as cholesterol ester, and rest 1/3 will be present as free cholesterol molecule. And note that cholesterol ester is the storage form of cholesterol. Now the cholesterol in its free form, it has got hydroxyl group at the third carbon, and the rest of the body of the cholesterol is hydrophobic. So that means cholesterol, it is an amphipathic molecule, and it is an important component of the cell membrane. In fact, it is an essential component of a cell membrane, because cholesterol, it is going to regulate a fluidity of the membrane. Membrane fluidity is maintained by a cholesterol. That means cholesterol do not allow membrane to become too rigid or too fluid. By doing so, cholesterol is going to regulate permeability of the membrane, and that's one of the important function of cholesterol in our cell membranes. Not only that, cholesterol is a precursor for corticosteroids. It is a precursor for sex hormones like estrogen, testosterone. It is a precursor for bile acids and bile salts, which are acting as a detergents in the digestion and absorption of dietary lipids. And also it is a precursor for vitamin D. Now, although every cell in our body can synthesize cholesterol, but the additional need of cholesterol, we can get it from the diet. So the foods of animal origin are the important source of cholesterol for us. Example, egg yolk is rich in cholesterol, meat or any food that is animal derived has some amount of cholesterol. Now coming to a basic structure of cholesterol molecule. So it is shown here. So the basic cholesterol structure, it includes cyclopentanophenothrin nucleus, and you are seeing that here. It has got A ring, B ring, C ring and D ring. So there are 4 rings in a cholesterol molecule, and total cholesterol has got 27 carbon atoms, as you can see here. So 27 carbon atoms are shown here. Now all these 27 carbon atoms are derived from acetyl-CoA molecule. And also you need to note that the cholesterol at the third carbon, a free cholesterol, means not attached to any fatty acid, is referred as a free cholesterol. So the free cholesterol has got a hydroxyl group at the third carbon. That is the one which gives polarity to a cholesterol molecule. And also there will be a double bond between 5th and 6th carbon of a B ring, and the D ring contains a hydrocarbon chain. 8 carbon hydrocarbon chain is attached here. So this is how a cholesterol looks like. And as I said before, acetyl-CoA is the precursor of all carbons that are present in a cholesterol molecule, and the acetyl-CoA you must be aware that we get it from oxidation of glucose. We get acetyl-CoA from ethanol. We get acetyl-CoA from some of the amino acids, and also we can get acetyl-CoA from oxidation of fatty acids. So, acetyl-CoA is a final common intermediate in our metabolism, and especially when the acetyl-CoA is derived from glucose under well-fed condition, and that acetyl-CoA is all diverted into cholesterol biosynthesis. It will be diverted into fatty acid synthesis. We are going to look into how exactly acetyl-CoA is diverted into fatty acid synthesis as we move on. Now, a little more than half of the cholesterol of the body, around 700 milligrams of cholesterol per day, we synthesize in our cells. That means a de novo synthesis of cholesterol contributes little more than half of the cholesterol that is needed for our body per day, and the rest of the cholesterol, it will be provided in the diet. Virtually, as I said, every nucleated cell in human body is capable of synthesizing cholesterol, and in that, liver and small intestine is the one that is significantly synthesizing cholesterol, which contributes to around 10% of the cholesterol that is made in the body. And coming to the location of cholesterol biosynthesis, so you got to remember, cholesterol biosynthesis is divided into two subcellular compartments. One is in the cytoplasm, initial reactions going on in the cytoplasm, and later it will be going on in the endoplasmic reticulum. You got to remember the location of cholesterol biosynthesis. Now let's move on to see a cholesterol synthesis process, which can be divided into 5 steps. Step 1 is synthesis of mevalonate from acetyl-CoA. Step 2 is formation of isoprenoid units, which are 6 carbon molecules. And the third step is condensation of 6 isoprenoid units to make squalene. And the fourth step is cyclization of squalene to make a parent sterol called llanosterol. And the fifth step is formation of cholesterol from llanosterol. So these are the 5 steps that we are going to go into while we understand how exactly cholesterol is synthesized in our body. Make sure you remember these 5 steps. I am going to repeat one more time. Number 1 step is we are going to study is acetyl-CoA, which is converted into mevalonate. That is synthesis of mevalonate from acetyl-CoA. Second step is formation of isoprenoid units from mevalonate. Third step is condensation of 6 isoprenoid units to make squalene. And the fourth step is cyclization of squalene to make a llanosterol. And the fifth step is formation of cholesterol from llanosterol. Now let's get into the very first step in the cholesterol biosynthesis, and that is synthesis of mevalonate from acetyl-CoA. Now as you see here in the figure, 2 acetyl-CoA molecules will condense with one another to make aceto-acetyl-CoA. This reaction is going on in the cytoplasm. Now note that acetyl-CoA in fact is made in the mitochondrial matrix. Formation of acetyl-CoA itself, it is going on in the matrix of mitochondria, whereas synthesis of cholesterol is going on in the initial stages of cholesterol biosynthesis. It is going on in the cytoplasm. So it is kind of, there is a barrier there. So on the left side I am writing cytoplasm here. This is a cytoplasmic compartment where the cholesterol biosynthesis is going on. On the right side is the mitochondrial matrix. Okay, and this is where in the mitochondrial matrix we have acetyl-CoA. So acetyl-CoA is there in the matrix of mitochondria. Cholesterol biosynthesis is going on in the cytoplasm. So there is a barrier there, that is inner mitochondrial membrane, and then we have outer mitochondrial membrane, and then we have cytoplasm there. So how acetyl-CoA, which is made in the mitochondrial matrix, how it's going to move into the cytoplasm to go into cholesterol biosynthesis or to go into fatty acid synthesis. So how that is done? So the transport of acetyl-CoA out of mitochondrial matrix into the cytoplasm you must be aware of. That's another important concept that you should know, and also I have explained that in a fatty acid synthesis lecture. Let me briefly repeat that here. So acetyl-CoA in the matrix of mitochondria will combine with oxaloacetate as a part of TCA cycle to make citrate. And the citrate, whenever TCA cycle gets saturated, citrate gets into TCA cycle. So whenever TCA cycle is saturated, the citrate builds up, and the citrate, it is going to be transported out of mitochondria by a transporter called tricarboxylic acid transporter. And the tricarboxylic acid transporter brings the acetyl-CoA carbons, 2 carbons present in acetyl-CoA. So these carbons are brought out of the mitochondrial matrix into the cytoplasm. So citrate comes into the cytoplasm here, and in the cytoplasm citrate breaks open into oxaloacetate and acetyl-CoA, and that's how citrate is bringing carbons out of mitochondrial matrix into the cytoplasm. So acetyl-CoA carbons are coming out of mitochondria into the cytoplasm in the form of citrate. Remember that. That's the concept you should remember. How the citrate comes out? Citrate comes out of mitochondria into the cytoplasm by a transporter called tricarboxylate transporter, because citrate is a tricarboxylic acid. It has 3 carboxylate group, and the transporter is tricarboxylic acid transporter. Okay, so in the cytoplasm there is an enzyme called citrate lyase or citrate lyase, and this citrate lyase breaks this citrate into acetyl-CoA and oxaloacetate, and acetyl-CoA diverted into fatty acid synthesis. It is also can be diverted into cholesterol biosynthesis, and that's what we are seeing it now. That is how acetyl-CoA comes into the cytoplasm. Now the 2 acetyl-CoA molecules, they condense with one another to make aceto-acetyl-CoA, shown in the figure here. This reaction is catalyzed by thiolase enzyme. Now the aceto-acetyl-CoA will combine with one more acetyl-CoA to make 3-hydroxy-3-methyl-butyryl-CoA, and that is referred as HMG-CoA. Now if you carefully look at the carbons of HMG-CoA and number the carbons there. So the carboxyl carbon is number 1 carbon, and then we have number 2, number 3, 4, 5 and 6 carbons here. So the third carbon is also called as beta carbon, because second carbon is alpha, third carbon is beta. So the third carbon here is attached with the methyl group. Third carbon is attached with a methyl group, and also third carbon is attached with a hydroxyl group. So I have highlighted that here now. So since third carbon is attached with a methyl group and also hydroxyl group, that's why the name of the molecule that is written here is 3-hydroxy-3-methyl-butyryl-CoA, and that is what is HMG-CoA. Note that HMG-CoA, it is a 6 carbon molecule, that's why it is a glutaryl. Glutaryl means 6 carbon. Now what happens to HMG-CoA? HMG-CoA will be reduced into mevalonate, and mevalonate, look at the number of carbons in mevalonate, it is 6 carbons still. Okay, so in order to reduce HMG-CoA into mevalonate, there is a requirement of two NADPH plus 2H plus molecules. And how do you get NADPH plus H plus? The predominant source of NADPH H plus is a oxidative phase of pentose phosphate pathway. In that we have glucose 6-phosphate dehydrogenase and we have 6-phosphoglucanolactone dehydrogenase. These are the two enzymes which are in oxidative phase of pentose phosphate pathway, which will provide needs of NADPH H plus in reductive biosynthesis. Cholesterol biosynthesis is one of the reductive biosynthesis pathway. Just to recap the sources of NADPH H plus for you. So NADPH H plus are coming from, in fact, 5 enzymes. Let me write it down, all those 5 enzyme names for you. This is just a recap. So sources of NADPH H plus, one is G6PD, glucose 6-phosphate dehydrogenase, which is a pentose phosphate pathway enzyme. Second is 6-phospho 6-phosphoglucanolactone dehydrogenase, that's the second enzyme. Third is maleic enzyme, which is involved in the conversion of malate into pyruvate. And the fourth is cytoplasmic isocitrate dehydrogenase, which is involved in the conversion of isocitrate into alpha-ketoglutarate in the cytoplasm. So these are the 4 enzymes which are there in the cytoplasm. All of them will give NADPH plus H plus, and that NADPH H plus will go into reductive biosynthesis pathway, and one of the reductive biosynthesis in our cytoplasm is cholesterol biosynthesis that we are looking at now. Now let's move on to see a formation of isoprenoid units and also how these isoprenoid units will form squalene. So these 2 steps we will look at together. As you see here in the figure, mevalonate, which is a 6-carbon molecule, will undergo a series of phosphorylation process. In fact, there will be 3 phosphorylation process going on. So I am going to highlight that now. So in the first reaction there is an addition of ATP, and the second reaction there is addition of one more ATP, and the third reaction there is an addition of another ATP. That means 3 ATPs are broken down here sequentially, one after the other, into ADPs, and there will be phosphorylation of a mevalonate, and then followed by there is a decarboxylation process going on. So phosphorylation and decarboxylation, I would just write it that way. Now it's phosphorylation and dephosphorylation of mevalonate is the one that is converted into isopentenyl diphosphate. So this isopentenyl diphosphate is a 5-carbon molecule, as you can count the number of carbons here. So isopentenyl pyrophosphate or diphosphate contains 5 carbon and 2 phosphates. Okay, penta means 5, as you can understand from the term. You know, penta means 5, di means 2. So isopentenyl diphosphate, this is what is the isoprenoid unit containing 5 carbon. Now this isopentenyl pyrophosphate or diphosphate, you can say it in 2 ways. Pyrophosphate means 2-phosphate, diphosphate is 2-phosphate. Some books will say isopentenyl pyrophosphate, some books will say isopentenyl diphosphate, both are correct. Now isopentenyl diphosphate will undergo isomerization reaction to become dimethyl allyl diphosphate. So one isopentenyl pyrophosphate is converted into dimethyl allyl pyrophosphate. So we have now two molecules. Now you got to look at like this. So in the cytoplasm, there will be multiple cholesterol biosynthesis pathways going on. In one cholesterol biosynthesis pathway, we get one mevalonate, and that mevalonate, and one side you make isopentenyl pyrophosphate or diphosphate, right? And the other cholesterol biosynthesis, another mevalonate making another isopentenyl diphosphate, and that isopentenyl diphosphate goes into dimethyl allyl diphosphate. That means with two cholesterol biosynthesis pathway, on one side you get isopentenyl pyrophosphate, pyro means di, and then the other cholesterol biosynthesis pathway, we are going to get the same isopentenyl pyrophosphate converted into dimethyl allyl pyrophosphate or dimethyl allyl diphosphate. Okay, so we have dimethyl allyl diphosphate coming from another cholesterol biosynthesis pathway. Now both of them, because these are going on in the cytoplasm, both of them, they condense with one another. That means, so we have 5-carbon isopentenyl diphosphate, 5-carbon dimethyl allyl diphosphate, so 5 plus 5 carbon, so 2 isoprenoid units, they both condense with one another and make a geranyl diphosphate. And now this geranyl diphosphate is a 10-carbon molecule. So we have a 10-carbon unit here. So basically this 10-carbon geranyl pyrophosphate, it has 2 isoprenoid units. Now from the third cholesterol biosynthesis pathway, which is running simultaneously, multiple of them will run simultaneously. So from the third cholesterol biosynthesis pathway, we are going to take one more isopentenyl pyrophosphate or isopentenyl diphosphate and condense that with geranyl pyrophosphate or geranyl diphosphate to make a farnesyl diphosphate, which is also can be called as farnesyl pyrophosphate. Okay, so this farnesyl pyrophosphate, now it will have 15 carbons. Why? Because 10-carbon geranyl diphosphate combining with 5-carbon isopentenyl diphosphate, together we have 15-carbon farnesyl diphosphate or farnesyl pyrophosphate, either way you can call it as. Now we have this farnesyl diphosphate, which is 15 carbon. So here it is, farnesyl diphosphate. Now what happens to farnesyl diphosphate? Now the farnesyl diphosphate will combine with another farnesyl diphosphate, and that makes a 30 carbon squalene. Two molecules are coming together now. How do you get that? Another molecule of farnesyl diphosphate. That you get it from other cholesterol biosynthesis pathway. In this cholesterol biosynthesis pathway, as it is shown here, you get one farnesyl diphosphate. Another cholesterol biosynthesis pathway where all these reaction going on up to farnesyl diphosphate, which is in the cytoplasm, that is taken here and both are combined, and that is made into squalene, and squalene is a 30 carbon molecule, and that's how we have made a 30 carbon squalene here. Alright, so this is what is the formation of isoprenoid units and formation of squalene. So we came all the way up to 30 carbon squalene. Next, let's move on. What happens to this squalene? Now the squalene will undergo ring closure to form lanosterol, and then lanosterol will undergo further reactions to make cholesterol. These reactions will be going on in the endoplasmic reticulum. Now your cholesterol biosynthesis is moving to the endoplasmic reticulum. Now let's see the details in the form of figure. As you can see here, in the figure, the numbered positions are those of the steroid nucleus, and the open and solid circles indicate the fate of each of the carbons in acetyl moiety of acetyl-CoA. So it is a complicated figure, as you can see, but by this time we have an understanding already. We came all the way from 2-carbon acetyl-CoA into complexity. This is what is the beauty of biochemistry, where majority of the complex molecules are made from simple things. A simple carbon, as 2-carbon acetyl-CoA, acetyl-CoA is just a 2-carbon molecule, and that 2-carbon acetyl-CoA, it is going towards making one of the most complex molecule in our cell is cholesterol, which is a rigid molecule containing 27 carbons. So that entire thing is shown here, figure that is given here in this particular slide. So let me just take you all the way towards where we stopped in the last slide, and then I will continue from there. Just as a recap, okay. So acetyl-CoAs, 2 acetyl-CoAs combining with one another to make aceto-acetyl-CoA, and that aceto-acetyl-CoA combining with another acetyl-CoA molecule to make HMG-CoA, that is 3-hydroxy-3-methyl-glutaryl-CoA, and the HMG-CoA undergoing a reduction process by HMG-CoA reductase enzyme, which is the most regulated enzyme in cholesterol biosynthesis, making mevalonate. And note that mevalonate is a 6-carbon molecule, and mevalonate now, and by that time already we have used 3 acetyl-CoA. Remember, 1 acetyl-CoA, 2-carbon, acetyl-CoA condensing with another acetyl-CoA, making aceto-acetyl-CoA, that is 4-carbon. So acetyl-CoA combining with another acetyl-CoA to make aceto-acetyl-CoA, that means 2 plus 2 carbon, 4 carbon, and that aceto-acetyl-CoA combining with one more acetyl-CoA to make HMG-CoA, and HMG-CoA has got 6 carbons, and that HMG-CoA is undergoing a reduction process done by HMG-CoA reductase enzyme, the most regulated enzyme in the cholesterol biosynthesis, making mevalonate, and mevalonate has got 6 carbons there, and that mevalonate is undergoing a series of phosphorylation process and also decarboxylation process. That's where one of the carbon is taken out of that 6-carbon mevalonate to make isoprenoid units. And the isoprenoid unit is isopentenyl pyrophosphate. So the isoprenoid units that are made here is isopentenyl pyrophosphate, isopentenyl PP, I'll write it as PP for pyrophosphate or diphosphate. And then isopentenyl pyrophosphate is going to undergo isomerization in another cholesterol biosynthesis pathway to make dimethyl allyl pyrophosphate. So one is 5-carbon and the other is 5-carbon. And now this isopentenyl pyrophosphate combining with dimethyl allyl pyrophosphate, both of them combine together to make geranyl pyrophosphate, which is a 10-carbon molecule. So they both combine and make a 10-carbon geranyl pyrophosphate. And the geranyl pyrophosphate will combine with one more isopentenyl pyrophosphate. So isopentenyl PP pyrophosphate, 5-carbon. So they both combine now to make farnesyl pyrophosphate, and farnesyl pyrophosphate is a 15-carbon molecule. Now this farnesyl pyrophosphate 15-carbon molecule, in fact it has, so the 15-carbon farnesyl pyrophosphate, in fact it contains 3 molecules of isopentenyl pyrophosphate in different forms. That is, 1 molecule of isopentenyl pyrophosphate, that is 5-carbon, which is converted into dimethyl allyl pyrophosphate, that's another 5-carbon. So 2 isoprenoid units there. So they both combine to make geranyl pyrophosphate, 10-carbon, 2 isoprenoid units, and 10-carbon geranyl pyrophosphate combining with one more isopentenyl pyrophosphate, 5 more carbon, that is 15-carbon farnesyl pyrophosphate. That means we have 3 isoprenoid units there in 15-carbon farnesyl pyrophosphate. So another 15-carbon farnesyl pyrophosphate combines with this. So 15 more carbons, that means 3 more isoprenoid units. So together we have 30-carbon squalene, that is a squalene there. And that 30-carbon squalene, it has 6 isoprenoid units, because this is how it came. isopentenyl pyrophosphate 5 carbon and then dimethyl allyl pyrophosphate 5 carbon making geranyl pyrophosphate 10 carbon. So 2 isoprenoid units there, 1 isoprenoid is 5 carbon. Okay. And then geranyl pyrophosphate combining with an isopentenyl pyrophosphate making 15 carbon, that means 3 isoprenoid units by that time, and 15 carbon farnesyl pyrophosphate combining with one more farnesyl pyrophosphate making 30 carbon squalene. Total we have 6 isoprenoid units. So that is what is written here, 6 times 6, and we made a squalene. So it is this squalene moving into endoplasmic reticulum, where a squalene epoxidase enzyme, it is going to, and followed by oxidosqualene lanosterol cyclase enzyme. So where there will be ring closure. So these are the two enzymes where squalene epoxidase enzyme, which is reducing the molecule, and ring closure happens with oxidosqualene lanosterol cyclase. So overall making a first sterol or the parent sterol called lanosterol, and this is how the lanosterol formation is going on. Alright, so once you get a lanosterol will undergo further reaction. Again there will be multiple reduction and decarboxylation reactions are going on, where NADPH, H plus molecular oxygen and carbon dioxide released, and that makes initially zymosterol, and zymosterol will undergo isomerase catalyzed reaction and followed by a reduction reaction, and then another reduction reaction done by reductase enzyme, ultimately making cholesterol molecule. So a multiple reduction and decarboxylation reactions will take lanosterol into cholesterol. I would say that way. Alright, so this is how we have made a complex cholesterol molecule which has got 27 carbon atoms. Okay, so let me just quickly revise all the things that we have seen and in a simple way. So listen to me carefully. 2 acetyl-CoA molecules combining with one another to make acetoacetyl-CoA. That's how carbons of acetyl-CoA coming into cholesterol biosynthesis. So HMG-CoA molecule undergoes reduction process to make mevalonate, done by HMG-CoA reductase enzyme, most regulated enzyme in the cholesterol pathway. Mevalonate undergoes phosphorylation and decarboxylation process to make isoprenoid unit, that is isopentenyl pyrophosphate, and the 6 isopentenyl pyrophosphate in different ways, like overall 6 isoprenoid units. So in that we have isopentenyl pyrophosphate converting into dimethyl allyl pyrophosphate. They both combine to make geranyl pyrophosphate, and geranyl pyrophosphate brings in one more isopentenyl pyrophosphate to make farnesyl pyrophosphate, and the farnesyl pyrophosphate attached with one more farnesyl pyrophosphate making a 30 carbon squalene. So like this 6 isoprenoid units are combined to make a squalene. I want you to know the names, at least remember the names of these intermediates here, that is geranyl pyrophosphate, farnesyl pyrophosphate, because geranyl pyrophosphates and farnesyl pyrophosphates, although they are going into cholesterol biosynthesis pathway here, but there are other uses of geranyl pyrophosphate and farnesyl pyrophosphates. So they go into other biological reactions. That's why we need to know them. We are going to go to that later. Now once we get squalene undergoes reduction and ring closure process to make lanosterol in the endoplasmic reticulum, and lanosterol further undergoes reduction and decarboxylation process to make a cholesterol. So this cholesterol, free cholesterol, that is made in the endoplasmic reticulum, so it will undergo a esterification process at its third carbon hydroxyl group. As you see here, there is a hydroxyl group at the third carbon, and this hydroxyl group will be attached by a fatty acid by a ester bond to make a cholesterol ester. So the cholesterol will be converted into cholesterol ester after attachment of a fatty acid. And the cholesterol ester is a storage form of cholesterol. And also this is cholesterol ester, this is how it will be transported out of the liver into the peripheral tissues in the form of VLDL. And VLDL will be metabolized into IDL into LDL. Ultimately, LDL is the one that carries cholesterol to peripheral tissues. Now, cholesterol attached to the fatty acid to make cholesterol ester. The type of fatty acid that is attached to the cholesterol is a linoleic acid, which is an essential fatty acid most of the time. Okay, and the esterification of cholesterol into cholesterol ester, it will be done by an enzyme called ACAT, that is acyl-CoA cholesterol acyl transferase, which is there in the endoplasmic reticulum. So this is how cholesterol is synthesized in two subcellular compartments. One is in the cytoplasm and other is in the endoplasmic reticulum. That's about a cholesterol biosynthesis there. Now, let's move on to see what are the derivatives of farnesyl diphosphate, which is also referred as farnesyl pyrophosphate. So, the farnesyl pyrophosphate or farnesyl diphosphate will be diverted into a special product formation in our cells, and one of the special product that is formed by farnesyl pyrophosphate is ubiquinone. Ubiquinone is also referred as coenzyme Q10 because it will have 10 isoprenoid units attached, and the important function of coenzyme Q10 is it is a mobile complex in the electron transport chain, and the amount of coenzyme Q10 that is found in the electron transport chain, especially which is located in the inner mitochondrial membrane, it all depends on what is the level of electron transport chain that is going on in individual tissues. A tissue with highest energy requirement will have highest quantity of coenzyme Q10 in their inner mitochondrial membrane. Example of such tissues is heart, neurons, liver and kidney. And one of the clinical application that you should be aware of here is whenever a patient or a person is prescribed with HMG-CoA reductase inhibitors to reduce their cholesterol levels. So, the HMG-CoA reductase inhibitor, it is going to target the rate limiting and the regulated step in the cholesterol biosynthesis, and that is conversion of HMG-CoA into mevalonate. So, thereby decreases the mevalonate formation. So, let me write down so that we understand the process properly. So, acetyl-CoA forming HMG-CoA. So, I will just write it, 3 acetyl-CoAs will form 1 HMG-CoA. HMG-CoA converted to mevalonate, and mevalonate will be going into farnesyl pyrophosphate. I will just write it as FPP, farnesyl pyrophosphate. Farnesyl pyrophosphate go all the way to cholesterol. This is just a simple, just a brief outline of cholesterol biosynthesis. Now, this farnesyl pyrophosphate can also go into ubiquinone formation, and farnesyl pyrophosphate can also go into 𝗏 formation, which we are going to see in the next slide. Okay, so ubiquinone, it is part of electron transport chain as we are seeing now, and 𝗏 it is used in N-linked glycosylation. So there are variety of proteins that needs N-linked glycosylation process. Now what is the application here? So the application is HMG-CoA reductase inhibitor. HMG-CoA reductase, that's the enzyme which is converting HMG-CoA into mevalonate, and the HMG-CoA reductase inhibitors like statins, so they are going to block the conversion of HMG-CoA to mevalonate. So what will happen? So in the presence of statins, there will be decrease in mevalonate formation, because ultimately we want to decrease cholesterol by giving statins So of course cholesterol levels will decrease because mevalonate is decreased. So we have achieved our intention there. So we have decreased cholesterol by targeting HMG-CoA reductase. But what happened there? Even farnesyl pyrophosphate is also decreased, because mevalonate decrease means farnesyl pyrophosphate is decreased. That's why cholesterol is decreased. So if the farnesyl pyrophosphate is decreased, that will decrease ubiquinone also. And if the ubiquinone is decreased means even electron transport chain will decrease. And the electron transport chain decrease means ATP levels will decrease. And imagine a tissue that needs highest energy, like even skeletal muscle, when it is contracting, it has to run electron transport chain. Heart, neurons, liver, kidney, which needs electron transport chain. In fact, they have got high concentration of coenzyme Q10. And also because FPP, farnesyl pyrophosphate decreased, means even 𝗏 is decreased. That means N-linked glycosylation is decreased. So this can, because of the decrease of ubiquinone, 𝗏, all of that, the consequences of decreased coenzyme Q or 𝗏 may amounts to the side effects. So many side effects that we see with the use of statins. Although statin is one of the best drug to decrease cholesterol levels in our body, especially in hypercholesterolemia conditions, but it comes with so many side effects, and that is possibly explained by lower levels of ubiquinone, lower levels of 𝗏 in which is done by use of statins, because it is targeting HMG-CoA reductase and decreasing mevalonate. Alright, so one of the effect of that can be seen over skeletal muscle as myopathy and rhabdomyolysis. So patients will show myopathy and rhabdomyolysis when they are on, some of them, not every patient. So some of the patients are very sensitive to this and present with myopathy, means muscle pain and breakdown of the skeletal muscle, and this can be prevented or like controlled to certain extent by supplementing coenzyme Q10 supplementation. Just have an idea about this, and it's an application of means applied aspect with the cholesterol biosynthesis pathway. As I said before in the previous slide, 𝗏 also play a role in post-translational N-glycosylation of proteoglycans and glycoproteins. Almost every protein in our cell or plasma is a glycoprotein, with very few exceptions. And the proteoglycans present in the extracellular matrix, all these needs N-linked glycosylation, and in fact N-linked glycosylation is the most common type of post-translational carboxylation process. Out of carboxylation, which are of two type of carboxylations, O linkase and N linkase, and out of O and N linkase is the most common one, and that can be affected in HMG-CoA reductase inhibitors use. So that can lead to complication, cut this. So it will lead to adverse effects. Now what is the other use of farnesyl diphosphate? It is the protein prenylation. So protein prenylation is, it involves the transfer of farnesyl or geranyl moieties, that is farnesyl pyrophosphate, geranyl pyrophosphate. So this farnesyl and geranyl molecules can be transferred to C-terminal amino acid cysteines of a target protein. So this process we call it as geranylation or farnesylation. So if the geranyl moiety is added, it is geranylation. If the farnesyl moiety is added, it's called as farnesylation over a protein molecule. So together we call it as protein prenylation. And the protein prenylation, it is facilitating protein-protein interactions, and also it is facilitating protein membrane interaction, where a geranyl moiety or the farnesyl moiety helps in the anchoring of certain proteins over the cell membrane. So protein-protein interaction, protein membrane interaction needs this protein prenylation, and the examples is like RAS and RAB proteins, Okay, and also there are certain drugs which have been under clinical trials which will alter this farnesylation and germinylation process. So we have farnesyl transferase inhibitors which are under clinical trials for treatment of certain cancers. So this is all about cholesterol biosynthesis and some of the derivatives of intermediates in the cholesterol biosynthesis. I hope this lecture has helped you in understanding a complex biochemical pathway, that is cholesterol biosynthesis. Thank you.